Thanks everybody for coming. So yeah, as Lena introduced, this is results from the first year of a potentially 3 to 5 year project that we have ongoing at Ames and Ceasing, and probably an eye-opening experience for the challenges we have as we start to think about interventions, because this experiment's working with Pocillopora, historically Damicornis, now known as acuta, and it's the fastest growing species, fastest species to reproductive maturity, and it's going to take us probably 5 years to get to the 3rd generation. So the reason we're here and thinking about actually intervening on a system as large as the Great Barrier Reef is because the Great Barrier Reef is dramatically threatened. This is an image of the bleaching severity that we saw at Russell Island off of Innisfail in 2017. Ubiquitously, species we thought were tolerant are bleaching and responding to the rate of climate change, and corals aren't keeping up with the rate of climate change. And we're seeing more severe, more frequent coral bleaching events. That's leading to a breakdown of the benthic community and the ecosystem that species like turtles, fish, and everything else that reefs support. These are images throughout the GBR following 2017 of widespread mortality. And the method that we're trying to use in this experiment is something that we have termed transgenerational plasticity, and Carly did a very good job at describing how the environment forms the tolerance of the parent and forms the tolerance for future generations. So the theory behind this project is basically that the parent, based on its environment and based on its local thermal range, has a certain temperature tolerance in a range that it is successful and can sustain. The theory here is that if we expose that parent to sublethal stress and push it to the outer edge of its tolerance levels, in 2, 3, or 4 generations, we think we can enhance and spread the thermal range that these corals are able to cope with. So the project now is basically, we've collected the parents, we've settled the larvae, and we're now growing this. We're growing 2 cohorts of the 1st generation. We have a cohort that were settled as soon as the corals came in from the reef. We call this the unconditioned cohort, and that's the cohort that I'm going to show you the tolerance traits today. We have also settled a second 1st generation cohort from those parents after they were exposed to sublethal stress for about 4 months, but those data are still to come as those juveniles grow up. So this is, the experimental design has used real-world models from the central GBR where these corals were collected. We've looked at, the blue line there is our control, and this is the 30-year average daily temperature range that these parents were exposed to and grew up in and survived 2 bleaching events. And then we're pushing the parents to, the medium treatment is a mid-century target, simulating 1.5 degrees of annual warming with 2.5 degree heating weeks, the equivalence of 2.5 degree heating weeks. A heat stress that we know causes bleaching. The end of century treatment is 5 degree heating weeks in the summer with 2 degrees of annual warming. And we hope that, and we think that, and we're testing whether the annual range and exposure to 1.5 and 2 degrees of warmer conditions will enhance the tolerance of the offspring. This is combined with, because as carbon dioxide enters the ocean, it's heating the ocean, but it's also acidifying the ocean. And so this is combined with present day 400 parts per mil PCO2. Mid-century is 685, and end of century is 900 parts per mil. Targets that are mid-century and end of century relevant. And the question here is, does preconditioning to this level of sublethal stress enhance tolerance? The Ceasing is a remarkable place that allows us to think about running long-term 5-year experiments like this. And we're using the system. You can see here we've got 2 shallow larval rearing tanks with a deeper adult, larger tank to separate the parents from the first generation and keeping those gene pools separated. We're running seasonal profiling and temperature like you just saw, daily variation in temperature and PCO2 that naturally happen with light. We're running with the artificial light systems natural seasonal sun changes in sunrise and sunset, and we have a lunar cue in here as well because we're dependent on the reproductive cycles. The lunar cue is keeping in time. So there's diurnal variation in temperature, there's seasonal variation in temperature and acidification, and it's keeping things as natural as we possibly can. And like I said, so we've collected these parents from 3 reefs. We collected from a reef near Cairns, a reef off of Innisfail, and a reef just north of Townsville, Coates Reef, Feather Reef, and Rib Reef. We started with 90 parents. We actually ended up getting 41 of them to release larvae, so that's our starting parent population experiment. We settled 7,000 unconditioned larvae in the first month, and following 1-year survival, we have about 4,000 F1s that have gone through the first summer heat stress across all treatments. And the traits of tolerance that we're using to assess how this preconditioning is influencing corals are basic things like survivorship, photosynthetic productivity, so we isolate the individuals in these incubation chambers and look at how the photosynthetic processes in respiration and calcification are changing across the treatments. So again, this is survivorship at the end of the first summer. We've got treatment across the bottom, present day temperature, mid-century and end of century, and then we have it split by reef. And the first thing that jumps out at you is we have pretty good survivorship, even at 5-degree heating weeks and 900 parts per mil PCO2, which is quite severe acidification stress. And we also have a location effect. So we've got quite strong survivorship from Coates Reef, and we'll get to that in a little bit. What's also interesting is that there's consistency in the genotype. So we have an obvious location effect on survivorship in the system. But when you split that by location, if you look at each individual grid cell there in this heat map, the darker the image, the higher the mortality of that individual genotype. And across in feather, the sensitive individuals under the present day ambient, what we thought shouldn't be stressful, are dying in ambient conditions, but they're also dying at the high end. So we're identifying tolerant and sensitive genotypes within the population with this experimental exposure. When you look at the physiology, this is split. We did incubations at the start of the summer, and then we did incubations after the heat stress. Calcification in the light in the summer, temperature is actually helping calcification as you'd expect to a point, but as the summer unfolds, we start to see the bleaching effect in the community as calcification rates start to drop at the high-end treatments. And that's accentuated when you go into the dark. We're actually starting to see individuals start to dissolve under these experimental conditions. There's not a whole lot of change in the metabolic rate of these individuals at high temperature and high PCO2, which is interesting in itself. We'd expect the metabolic rate to go up with high temperature and high PCO2. But then when you start to look at the photosynthetic traits, this lines up really well with our survivorship data. Coates Reef at the end of, Coates Reef and Feather Reef at the start of the summer are saturating at about the same height. So anybody not familiar with these figures, we've got oxygen production on the y-axis as we increase the light intensity during a light curve, traditional PI curve. When you start to get into, so in the pre-summer, Coates Reef and Feather Reef are performing at about the same level. Rib Reef is already probably more of a dark adapted symbiont. But Coates Reef, after the heat stress and after the summer, are still outperforming the other two locations, which is evident in their survivorship as well. So what's different about Coates Reef? We tried to constrain the collection within a similar temperature range. There's only about a 0.3 of a degree difference between the annual temperature of Coates Reef and Rib Reef. But we did see more bleaching happen, so degree heat index in the top. There's not a big difference in heat exposure for 16 or 17 prior to collection, but we did see more bleaching at Coates Reef. So there is the chance that there was natural selection at Coates Reef, that we were already collecting the most tolerant individuals from Coates Reef because there was potentially higher mortality at that location. So just to summarize, sublethal stress, both naturally following a bleaching event and in an experiment like this, fairly quickly identifies tolerance within the population and sensitivity within the population, which can definitely enhance your fragment and outplant restoration type efforts. Reef and parental genotype definitely influences tolerance, and as we work through this experiment, hope to understand that quite a bit better. And preconditioning the parents and the larvae, like I said, can definitely enhance selection of tolerant individuals prior to field deployment. So the next steps in this experiment are to repeat the same physiology experiments on the conditioned F1 cohort, and then hopefully by about this time next year we should be to the second F2 generation, again to understand whether preconditioning is enhancing the tolerance of these individuals. And this is a joint project between Ames and Ruth Gates' team in Hawaii. They're doing fairly similar things with the Hawaiian Pocillopora, so I'd like to thank Ruth and Ariana Hoffmeyer, who are working closely with Madeleine for the ideas, and again, definitely Paul Allen Philanthropies for the large amount of funding that's supporting this work.
We have time for questions. Excellent.
Mel,
What type of symbionts did you have?
we're testing that. So part of the molecular work and the epigenetic sides of the steps are now to, because these are asexually produced clones, we're testing the clonality in the population from each of the reefs and looking into the symbiont,
Do you
differences in the
think that
symbionts.
there are any between the reefs?
Based on that PI curve, I would expect that there's probably differences in the symbiont population. There's pretty big differences there.
Cool. Thank you.
Anyone else? You must be not just settled in like that audience. Thank you very much.